Welcome to chapter 7.2. We're going to talk all about catabolism in this section and why we call it the microbial buffet. But before we start into 7.2, let's do a review question. So review question number 1, the hydrolysis means, lysis, remember, means break. Hydro means water, so we're going to break with water. The hydrolysis of a protein into amino acids is an example of A, anabolism, B, catabolism, C, lithotrophy, or D, nitrogen fixation. Okay, so pause the video, flip through your notes, and see if you can find the answer. Okay, the answer to this is B, catabolism. So whenever we're breaking bonds, right, lysis means to break. So we're taking a protein, which is a large molecule, and we're breaking it into smaller molecules, aka amino acids. But this is the definition of catabolism. So the answer is B. Now, if it was the reverse, so if we were taking amino acids and making a protein, this would be anabolism. Okay, so in 7.2, we're going to talk about the microbial buffet, so how microbes use food molecules to make energy. So the section objectives is we're going to be able to describe how catabolism converts many complex food molecules to a few kinds of catabolites. So catabolite is what we break these food molecules into. Explain how catabolism yields energy and how the energy is stored for use. And you need to be able to describe how the energy carriers ATP and NADH transfer energy between energy yielding and energy spending reactions. Okay, so we're going to talk about what happens when we break things down. Okay, so just a reminder, catabolism is the breakdown of complex, aka big, organic food molecules into smaller components. So we talked about our different macromolecules, so carbohydrates, lipids, proteins. These can all go through catabolism. They can all be chemically broken down. Now, when we're breaking things down, these reactions are what we call oxidative, and we'll talk about what that means here in a second. Okay, but the main thing that comes out from this is when we break these molecules down, every time we break bonds, we release energy. So we're going to release energy in the form of high energy electrons, and that's going to carry this energy from one molecule to another. So we're going to release the energy from breaking these things like carbs and lipids down, and we're going to take that energy and we're going to transfer it to ATP, adenosine triphosphate, our main energy currency for the cell. Okay, and because we're breaking down big molecules like carbohydrates, lipids, and proteins, we're going to produce smaller biomolecules. So from proteins, we're going to create amino acids. From lipids, we're going to break them down into fatty acids. And carbohydrates, we're going to break them down into simple sugars like glucose. Okay, so we need to know what catabolism means. Okay, so this leads to the question, what can we catabolize? Now, what an organism can catabolize is all dependent on the types of enzymes that organism has. So let's talk about me and you. Okay, we can break down starch into glucose, and we can then use it for cellular respiration. Now, for starch, this is a starch molecule up here. Okay, so we have our starch. Okay, so if you can see all these little starch or glucose molecules have the CH2OH group on the top all the time. Okay, so we have enzymes that can go through and break these down. They're called amylases because this is an amylose chain. That's what we call it. So we can break this amylose chain down into individual glucoses that can then be used for catabolism. Okay, we have a similar polysaccharide that we talk about in Biology 201, which is cellulose. Okay, so cellulose is down here. Now, if you notice up here, all of our CH2OH groups are on the top on this molecule, but on cellulose, every other one, so like this guy, they're up just like with starch, but this guy and this guy, they're pointing down. Okay, this is a really simple change, right? Instead of having them all up, we alternate. So one up, one down, one up, one down. You wouldn't think this would be this wouldn't change anything, right? It's still made of glucose particles, or sorry, molecules, not particles. So the thing is, though, when we switch that cellulose bond by 180, when we rotate that glucose by 180, this means that our enzyme that normally breaks down starch, it no longer has the shape needed to break down cellulose. So humans, we can't break down cellulose. Most animals can't. In fact, we're going to depend on a symbiotic relationship with bacteria and well, mainly for us bacteria, that's going to break these down for us. Okay, so even though these two molecules are made of the same thing, because we flip one of those bonds upside down, it means that we can't break it down, we can't use it. So we're going to be dependent on the bacteria in our intestinal tract in order to do this for us. Okay, so things like celery, which are mostly water and cellulose, we don't get a lot of nutrition from them because we can't break them down. We have to depend on the bacteria in our guts to do so. Okay, so catabolism is going to break different things down, and what you catabolize is all going to depend on what type of enzymes you have. Now, the really cool thing about a lot of living organisms is that no matter what you start to catabolize, you're going to send that catabolite into a central pathway. So most organisms, us, bacteria, so many different things, if we start out with carbohydrates, so we have our polysaccharide, we're going to start to break that down into smaller pieces until we have individual glucoses, okay, so individual monosaccharides. Well, for us, we're going to take that glucose and we're going to put it into a process called glycolysis. Glycolysis, lysis means break, glyco means sugar. We're going to literally break glucose down. Okay, so there's so many organisms that do glycolysis. This is why it's called a central pathway. Okay, so once we add that glucose in, we're going to break it down into pyruvate. So where you go from here depends on what kind of conditions you're in. So if you're an anaerobic microbe, you are going to do fermentation. So you're going to come over here to the fermentation pathway. And depending on what type of fermentation, you're going to make different products. Okay, so for us, we're going to go through respiration. So our pyruvate is going to go and be converted to acetyl-CoA. And this acetyl-CoA is going to go into the next step of the central pathway, which is the citric acid cycle. Okay, sometimes we refer to this as the TCA cycle or the Krebs cycle. Now, I'm going to use Krebs cycle more often than TCA cycle, and I'll use TCA way more than the citric acid cycle. But no matter the name, it's the same thing. Okay, the whole point of this central pathway is to break glucose down carbon by carbon and harvest as much energy as we can from the breakdown of sugar. Okay, so we can start with lipids. Okay, we can break it down to glycerol and fatty acids. The glycerol is going to enter into glycolysis, just like glucose, and it's going to start the process there. The fatty acids can directly enter this reaction here and be converted to acetyl-CoA. Different bacteria and things like termites, they're going to break down lignin in wood. Okay, this is a hydrocarbon, it's an aromatic. Okay, so it's going to start to break down into simpler components from lignin, and that's going to go back to this acetyl-CoA step. Okay, so proteins, same thing. We're going to break down the peptide chain into individual amino acids, and it's all going to enter this acetyl-CoA step. Now, just like with carbohydrates, all of these starting materials can then go to fermentation or they can go to respiration, depending on which pathway is needed for the organism. Okay, so the overall theme of this catabolism pathways is that we are going to break down everything into high energy electrons and then take that energy and store it into a molecule made to store that energy, mainly ATP. Now for this class, we are going to focus on glucose catabolism. Now because that's what we're most familiar with. All these are different pathways, and every time you see an arrow these arrows in this pathway are controlled by an enzyme. Okay, so overall, no matter where you enter into this pathway, the overall reactions are pretty efficient and they work very well. Okay, so let's talk about this energy transfer. So what is actually happening? So the reactions that are going to control this energy transfer and metabolism are what we call oxidation reduction reactions. We're going to refer to them as redox reactions. Okay, so these are two reactions that have to occur together, and they're going to help transform matter into energy and then going to store into the bond at some point in the reaction. So each step in a redox reaction has its own characteristics. So with oxidation, we're going to take our substrate or our reactant, and we're going to strip it of electrons. So it's going to say, we're going to say it donates or loses electrons. So it's going to give up the high energy electrons. So when a substrate is being reduced, okay, this means that that substrate is accepting or gaining those electrons that we took in the oxidation reaction. Okay, so in terms of catabolism, the biomolecule that we are breaking down, this is going to donate the electron, so it's going through oxidation. Okay, so the electron carriers, these are intermediates that are participating in the reaction, so they're going to be what carries these high energy electrons through this whole pathway. Okay, they're going to accept the electrons to carry that energy. So when we're talking about energy transfer, we're talking about the transfer of electrons from one molecule to another. Okay, so we're going to talk mostly about ATP and NAD, specifically NADH in this lecture. So NAD is the oxidized form. So when it gains electrons, so when it's reduced, it becomes NADH. Okay, so different carriers can carry different amounts of energy. So NADH has 3 times as much energy as one molecule of ATP. Okay, the thing is with redox reactions, because they are coupled together, one of them is going to be the catabolic reaction, and it's going to then provide the energy to drive the anabolic reaction, so the flip side of the coin. Okay, so with redox reactions, we have some confusing terminology. So if it's being reduced, we are gaining electrons. If we were being oxidized, we are losing losing electrons in the reaction. Okay, so we're looking for this transfer of electrons. Okay, so one of the ways that we remember this is with some simple mnemonics. So we have Leo goes grr. So Leo the lion says grr. So Leo, loss of electrons is equal to reduction. Gain of electrons is equal to reduction. Okay, or you can remember it as Leo goes grr. So, oh, it's so nerdy, even for me. Okay, same thing. Loss of electrons is oxidation. Gain of electrons is reduction. So I am a chemist by trade. We learn oil rig when we're learning about redox reactions. So oxidation is the loss of electrons and reduction is the gain of electrons. Choose whatever mnemonic works for you. You just need to know what each one means. These will be things that you'll be tested on. Okay, mostly we're going to look at some diagrams and you'll have to tell me what's going on. Okay, so let's look at some of these. Okay, so we have to take this energy from breaking these molecules down so that we can use that energy later. So we have to be able to store that energy. Okay, so we are going to store the energy into a molecule called ATP. Now, when we're going to make ATP, we're going to start with ADP. So ADP stands for adenosine diphosphate. Okay, so we're going to have this adenine molecule here. So this ADP. Okay, so adenine ribose, that's adenosine. And then we're going to have these 2 phosphate groups. So what's going to happen is the energy in the electrons, we're going to take this phosphate group here, and we're going to add it to this chain of phosphates with the energy from the electrons, so that the energy is going to be stored in this bond that we're making, and it's going to form ATP. Okay, so when we are using these phosphates, these bonds are our energy storage. Okay, so they're what are known as high energy bonds. Okay, so we're going to store the energy when we make them, but when we go to break them later, they're going to release a lot of energy. Okay, so one of the things that we say is that ADP is going to be the low energy state of the molecule. Okay, so when we add the phosphate and the electrons that are carrying the energy, this is going to make it the higher energy state. Okay, so each of these gammas are going to, each of these phosphates are going to have a designation. So the one that is closest to the ribose, so this little home plate looking thing, is going to be the alpha, beta, and gamma. So when we are using ATP to provide energy, what we're going to do is we're going to cleave or cut the bond between the beta and gamma phosphate, and that's going to provide energy to power reactions. Okay, but it's all because we're transferring energy from our catabolite, so whatever we broke down, to ATP. Okay, so when we break ATP down, we call it ATP hydrolysis. We are literally going to take a molecule of water, put it across the bond, and it is going to lyse or break the bond with the water. Okay, so when we break the bond between the beta and gamma phosphates, we're going to release quite a bit of energy in standard conditions. So things like with bacteria. So we'll release about minus 7.3 kilocalories per mole. Whenever you see negative in science, this means we're releasing energy. We made too much. Okay, we're not going to use it all. Okay, the thing is with ATP hydrolysis, in our bodies, we can actually double that amount, so around 14, 15 kilocalories per mole. Okay, the thing is with ATP is as a molecule, it's unstable, which is why it works so well to store energy in, is because it will hydrolyze quickly, and we'll have to take that ADP that's left and build it up back into ATP. So we always are going to have a place to store the energies from when we break down glucose. Okay, so with ATP hydrolysis is what we call an exergonic or energy releasing reaction, and it's always going to be coupled with an endergonic reaction, so a reaction that needs energy. The thing is if it's not coupled to a second reaction, all the energy that we made from breaking that bond between the beta and gamma phosphate is going to be lost as heat. Okay, so let's talk about electron carriers and how we transfer electrons. Okay, so this is NAD plus. This NAD is nicotinamide adenine dinucleotide. We're just going to call it NAD because that is a mouthful. Okay, so NAD is NAD plus is oxidized. Okay, so we've lost electrons. Okay, and it can be reduced, so we can add electrons and gain energy. Okay, so NAD plus can accept 2 electrons, and it's going to do this by way of 2 hydrogens. Okay, so we're also going to gain 2 hydrogens when we are reduced. Okay, so we're going to add those here. So we're going to get the H on the nicotinamide ring, which is this one with the N. Okay, so we're going to add an H here. That's going to take away the positive sign. And we're going to add the electrons there. And then we're going to have this H plus ion. This is why it's called NADH plus H plus, because of the second ion. Okay, so it's going to carry these 2 electrons and an H plus down through the pathway. So these electron carriers are going towards the final step of respiration. So for us, that's oxidative phosphorylation. There's something that's really important that's made in this reaction. So when we pull these electron carriers, pull the electrons off these carriers, we're also going to reach H plus. So we talked about earlier that proton motive force. This is where those hydrogens for that proton motive force that power things like ATP synthase are going to come from. Okay, so not only are we transferring energy through this whole pathway, we're also bringing hydrogens to build that proton motive force. Okay, so when we add, so NADH plus H plus is going to be the reduced state, which means we're going to carry those 2 electrons and the hydrogen throughout that 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 pathway. Okay, we have other electron carriers. We have FAD plus, FADH2, NADP plus, and NADPH. We'll talk about these in a later section. Okay, so we call NAD an electron carrier, and all these an electron carrier is because the oxidized form is going to accept electrons. Okay, so it's going to be reduced, and then we're going to have that NADH that can then be oxidized to release the electrons into the electron transport chain that's part of that oxidative phosphorylation step. Okay, so these electron carriers are a very important piece of this whole metabolism process. Okay, if we didn't have these, we didn't have a way to transfer the energy, so those high energy electrons from atom to, or from molecule to molecule to molecule. Okay, so that's important. Okay, so that is the end of chapter 7.2. We've talked about catabolism. We've talked about the central pathway. We're going to talk about that central pathway, so glycolysis and the Krebs cycle and oxidative phosphorylation in more detail. We'll also talk about fermentation in more detail. We need to remember that ATP stores energy in the bonds. Okay, so we're going to take those electrons and we're going to store them in that bond between the beta and gamma phosphate. Okay, NAD and NADH are going to act as electron carriers. So NADH is going to store energy that's going to be associated with that pair of electrons, and it's going to carry it through the pathway till we get to the electron transport chain, and then we will start to make the proton motive force, and we'll take those high energy electrons and start to store the energy in the ATP bonds. Okay, so that is it for chapter 7.2. We'll see you for the video for chapter 7.3.